Please subscribe to my channel and do hit the bell icon. All dear students, so in continuation to our topic, that is antimicrobials, so today we'll be discussing about a new topic, that is protein synthesis inhibitors. So basically these are antimicrobials whose main mechanism of action is to inhibit protein synthesis of the bacteria. Now, first of all, we shall discuss how proteins are synthesized in bacteria, and then we will discuss how antimicrobial can inhibit the protein synthesis. Now, for protein synthesis, message is brought by mRNA. That's why the name messenger. Now, these mRNA, they contains codons, complementary to which the amino acid will be placed in the proteins. Now, over this mRNA, two subunits of ribosomes, that is a larger subunit and a smaller subunits, will be attached. Now, in case of humans, this a larger subunit is 60S and the smaller subunit is 40S, whereas in case of bacteria, the larger subunit is 50S and the smaller subunit is 30S. Now, when the ribosomes are attached, now there is creation of two sites on the mRNA. These are P site and A site. P means peptidyl site and A means acceptor site. Now, amino acid chain which is already present is attached to the P site, and the new amino acid will attach to A site. So, suppose already we have got 3 amino acid, they will be attached to P site, and the new amino acid will be attached to A site. Now, this new amino acid is brought by tRNA, that is transfer RNA. So, transfer RNA which contains anticodon complementary to the codon on mRNA, only one particular tRNA will come. It will carry one particular amino acid. So, transfer RNA brings a new amino acid and it will get attached to A site. After the tRNA attaches, now there will be formation of a peptide bond between these two amino acids. Okay, so this is formation of peptide bond. Now peptide bond forms. Now all the amino acid from P side will dissociate and it goes to A side. So what happens is, now this P side and A side is created and all the amino acid has gone to A side. But we know that the tRNA, it attaches only to A side. But now A side is not free. So we want to make A side free. So what happens is, this ribosome, it will move forward. Okay. So when the ribosome move forward, the next site is exposed. So this site will become P site now, and the new site will become A site. So basically all of these are shifted back to P side. So when the ribosomes move, all of these are shifted to P side and A side becomes free. And this step is basically known as translocation. Okay, so what happens over here? Translocation occurs and all the amino acids are shifted to P side. So basically A side becomes free. So next what happens? Next a new tRNA comes and it will come and attach to A side. And in this manner the protein synthesis continues. So if a drug can inhibit any of these steps, it can be used as antimicrobial drugs. So next we shall check out the mechanism by which the drug can work. Okay. Now the very first way by which these antimicrobials can inhibit protein synthesis is, like we know that tRNA, it will come and it will attach to A site only. But if we can give a drug which will inhibit this attachment of tRNA on A side, means if tRNA do not attach to A side, then proteins cannot be synthesized. Okay, So this action is stopped by a drug that is tetracycline. Okay, so what tetracycline does, it will inhibit the attachment of tRNA on A side. Okay, so we know tetracycline, it is an antimicrobial. We know that it is a protein synthesis inhibitor. But what exactly tetracycline does, tetracycline will inhibit the attachment of tRNA on the A side. Next step of protein synthesis in bacteria was formation of peptide bond. Okay, so we know that these two amino acid, they will bind to form a bond that is known as peptide bond. So we can give a drug that will inhibit the formation of peptide bond, and if a drug inhibit formation of peptide bond, it will also inhibit the protein synthesis. Okay, so we have got a drug known as chloramphenicol. The main mechanism of action of chloramphenicol, yes, it is also protein synthesis inhibitor, but the main thing that chloramphenicol does is it will inhibit the peptide bond formation. Next, third step in protein synthesis of bacteria was to produce translocation. Okay, so we know that once this peptide bond is formed, then this ribosome will be moving, which is known as translocation, forming a new A site, isn't it? So this is translocation. But if translocation does not occur, protein synthesis will be inhibited, right? So we have got drugs that will inhibit translocation, and these drugs are macrolides, clindamycin, and quinpristine. Okay, so three drugs we have got, or three group of drugs we have got, that will produce action, that is inhibition of translocation, by which they will inhibit the bacteria. Now we can remember these three drugs from the mnemonic MCQs. Okay, MCQs means macrolides, clindamycin, and quinpristine, and their main mechanism of action is they will inhibit translocation. Next, the fourth way by which we can inhibit protein synthesis and kill the bacteria is by producing misreading. See, normally what happens, we have got codon on the mRNA, and complementary to which anticodon is present on the tRNA. Okay, and it will attach and it will produce protein synthesis, right? But now what we can do is, we have got a group of drug known as aminoglycosides. They will produce misreading of mRNA. Okay, so this misreading of mRNA is done by aminoglycoside. So basically what happens over here is, just suppose we have got AUG as the code on the mRNA at A side, so that tRNA will attach, which contains the complementary codon as UAC, right? No other tRNA can attach. So this will carry one particular amino acid only. When we give aminoglycosides, this code is read wrongly, means instead of A in AUC, it can be read as C. So when it is being read C, a wrong tRNA comes. This will contain a different anticodon. So when a wrong tRNA comes, it will carry a wrong amino acid. That means protein which we want to synthesize will not be synthesized, but rather a wrong protein is synthesized, and most of the time this wrong protein is a cidal protein, and it will kill the bacteria. So this is very much important to remember, that aminoglycosides are the only protein synthesis inhibiting drug which are cidal. Like most of the other protein synthesis inhibitors, they are bacteriostatic, but aminoglycosides, they will actually kill the bacteria. Okay, so these are the four mechanisms by which protein synthesis inhibitors can produce inhibition of the bacteria. Now we have discussed the mechanism of action of various protein synthesis inhibitors. Next thing to discuss is that on which ribosome it will act. Like in case 30s ribosome, on the smaller subunit, aminoglycoside and tetracyclines will be attaching, whereas on the larger subunits, the other protein synthesis inhibitors like chloramphenicol, macrolides, clindamycin, and quinpristine will be attached. Okay, now next we'll be discussing about the tetracycline antibiotics. Now, as the name says, their chemical structure contains four cycles. Okay, so tetracycline drugs coming under this group are: tetracycline is also a drug, oxytetracycline, chlortetracycline, demiclocycline, doxycycline, and minocycline. Mechanism of action is very simple. You know, again I am revising. We know that tRNA will come and it will get attached to A site. But what tetracycline will do in presence of tetracycline, this tRNA, it cannot come and attach to the A site, right? Next, we shall check out the uses of tetracycline, and it can be remembered from the mnemonic tetracycline itself. Okay, so TET is for tetracycline, R is for rickettsial infection. Tetracyclines are drug of choice for all the rickettsial infection. A is for atypical pneumonia. Nowadays, drug of choice for atypical pneumonia are macrolides, but tetracycline can also be used as an alternative. C is for cholera. Tetracyclines are drug of choice for cholera. Y is for Lyme's disease, like tick-borne illness caused by Bacterium borreliosis. Tetracycline can use for treating Lyme disease also. CL is for chlamydial infection, LGV. LGV means lymphogranuloma varianum, which is caused by chlamydia. Now these tetracyclines are drug of choice for this kind of infection. IN means inguinal granuloma. Tetracyclines are again drug of choice for inguinal granuloma. And lastly, E is for epidemics of plague, where again tetracyclines can be used. Okay, so basically tetracyclines, these are drug of choice for rickettsial infection, for cholera, for LGV, that is lymphogranuloma varianum, and also for inguinal granuloma. Okay, in other conditions it can be used as alternatives. Tetracycline are drug of choice means tetracycline group, and for most of these infections the drug of choice is doxycycline. Okay, next we shall check out adverse effects of tetracycline, and it can be remembered from the mnemonic Kapil Dev. Now K means kidney failure. So tetracycline are contraindicated in kidney failure except doxycycline. Why? Because it is excreted in bile. Rest all other tetracycline are excreted via renal route, therefore they are contraindicated. Next, A is for anti-anabolic effect. Tetracycline can produce anti-anabolic effect. Long-term tetracycline use can cause loss of muscle mass. P means phototoxicity. So these tetracycline are phototoxic. It can lead to rash if tetracycline is exposed to sunlight. Here, demiclocycline is highest phototoxic. I is for insipidus, that is diabetes insipidus. It can be caused. Tetracycline inhibits release of ADH hormone, therefore diabetes insipidus occurs. Here again demiclocycline have got highest risk of causing diabetes insipidus. L is for liver failure. So tetracycline are also contraindicated in renal failure. D is for dentition and bone problem. Tetracycline can affect the growing teeth and bone, therefore contraindicated in pregnancy and children. E is for expired tetracycline. Tetracycline should not be given after expiry date. Like other drugs, if we give after expiry date, normally it becomes ineffective. But problem with tetracycline is some side effects appears which are normally not present, like for example Fanconi syndrome caused only by expired or outdated tetracycline. Lastly, V means vestibular dysfunction. So tetracycline can produce vestibular dysfunction also, and highest risk is with minocycline. Okay, so these all are adverse effects produced by tetracycline. Now suppose this is a bacteria, okay, and whenever we give tetracycline, they inhibit the growth of bacteria. So after 2 days, means for 2 days when we give tetracycline, next for 5 days, 1 month, 6 months, for 1 year, and so on, we give tetracycline, and these basically they will inhibit the bacteria. So now these bacteria, they calls for a meeting with all the other bacteria. So what they decided that we will not tolerate this, means tetracycline is coming and killing us, we will not tolerate this. So they planned, and what they did, whenever tetracycline comes, they will kick off the tetracycline. Okay, so they have produced a kick. Okay, that means they, because of this kick, now they remove the tetracycline out of the bacteria. So what does it mean? It means that resistance has been developed to tetracycline, and the name of this kick is known as efflux pumps. Okay, therefore resistance to tetracycline develops because of efflux pumps. Now we know that bacteria has efflux pumps, and now it is resistant to tetracycline. So now we also become very much intelligent. So what happens? Upon bacteria, whenever tetracycline come, this efflux pump will kick off the bacteria. So now what we have developed? We have developed a cutter. Now we are cutting this leg only. And the name of this cutter is tigecycline. Okay. So mechanism of action and everything of this tigecycline is just like tetracycline, but chemical structure is different. Tigecycline is chemically belonging to a group of drug called as glycycline. Now the special point regarding this tigecycline to remember is it is resistant to efflux pumps. This we know. Next thing is it is very broad spectrum but not effective against pseudomonas. And lastly, it is secreted in bile, therefore safe in renal disease. Okay, so this is all about tetracycline. Next, we shall discuss about chloramphenicol. Now, regarding chloramphenicol, the mechanism of action is very simple. Like in presence of chloramphenicol, what happens means it will basically inhibit this peptide bond formation. Okay, so chloramphenicol, it is a protein synthesis inhibitor, and where exactly it will produce action means it is basically inhibit the peptide bond formation. Now, the special point to remember regarding chloramphenicol is chloramphenicol is rarely used nowadays. Okay, now in general, basically, why do why we do not like to use any drug in general means we have got basically two main reasons. Drug may not be effective and drug may be toxic. In case of chloramphenicol, we have got both the reason. Chloramphenicol is not very effective and chloramphenicol is very toxic. Okay, now we'll check out first thing, like why it is not that much effective. See, initially chloramphenicol, it was the drug of choice for enteric fever long back. Okay, because chloramphenicol, it was able to inhibit this enzyme salmonella, this organism bacteria salmonella long back. But now what happens, salmonella has become resistant to chloramphenicol. Okay, so bacteria has become resistant to chloramphenicol, so therefore they are not effective against most of the organism. This is the reason why the drug is not very effective. Next thing is that this chloramphenicol is very much toxic. Why? Because chloramphenicol can cause bone marrow suppression, high risk of causing bone marrow suppression. Therefore we do not use chloramphenicol because it is not that much safe. And secondly, it is contraindicated in newborn babies because it can result in gray baby syndrome. Chloramphenicol can cause cyanosis in baby, which is called gray baby syndrome. So because of these two reasons, we do not like to use chloramphenicol commonly. Then where exactly chloramphenicol is used, means what is the main use of chloramphenicol means nowadays chloramphenicol is mainly used in treating meningitis, particularly the meningitis where the bacteria is resistant to ceftriaxone. See, basically ceftriaxone is the main drug for treating meningitis, and if bacteria, if they do not respond to ceftriaxone means we can in place of that we can use chloramphenicol. Apart from meningitis, chloramphenicol is effective against anaerobic bacteria. And lastly, rarely, if sensitive, we can use chloramphenicol for treating enteric fever or typhoid fever. Okay, so this is all about chloramphenicol. Next, we shall discuss about another protein synthesis inhibitor, that is macrolides. Regarding macrolides, the mechanism of action, like after formation of peptide bond, there will be translocation taking place, isn't it? Now, macrolides, what they will do, they will inhibit this step of translocation. Not only macrolides, we have got MCQ drug, that is macrolide, clindamycin, and quinpristine, which will producing translocation. Now, the drugs coming in this group mostly ends with thromycin. Like we have got erythromycin, roxithromycin, clarithromycin, azithromycin. One more macrolide which is recently been used is fidaxomicin. It is new macrolide. Now fidaxomicin, it is mainly used for pseudomembranous colitis. Therefore, it is mainly indicated in mild to moderate pseudomembranous colitis. Next important point to remember regarding macrolides are, first of all, macrolides are second line drug to penicillin. Now regarding penicillin, we know they are basically effective against gram-positive bacteria, right? Normally we prefer penicillins. Why? Because they are cidal drugs. They are bactericidal drugs. But conditions when we cannot use penicillin, then at that time the drug of choice will be macrolides. Like for example, we know that a drug of choice for treating syphilis is benzamin penicillin G, right? But suppose if the person is allergic to penicillin, then the drug of choice for treating syphilis will become macrolides. Okay, so the major clinical use is second line drug to penicillin. Condition where penicillin cannot be used, at that time we can prefer macrolides. Now, apart from this, macrolides themselves are the drug of choice for certain conditions such as cancroid, legionella, atypical pneumonia, and pertussis, that is whooping cough. Now, these can be remembered from the mnemonic clap. Okay, so macrolides are drug of choice for all these conditions, and apart from these, they are second line drug to penicillin. Next, we shall check out the major adverse effect of macrolides, which can be remembered from the mnemonic, yes, macro. Now, M is for motilin receptor. So, these macrolides basically they stimulate the motilin receptor which is present in your gut. Now, what exactly is the role of this motilin receptor means? Suppose this is your GI tract and on the gut, this yellow star you can see, that is a motilin receptor. So, normally whatever is there in your GIT, it will be moving with its own speed, with its own pace. Okay, the gastric motility will be having some kind of time, some kind of activity, and with that speed, with that pace, the drug or the food will be moving into your GI tract. Okay. Now, what this macrolides will be doing is they are stimulating this motilin receptor, because of which the GI motility will be very much fast, leading to diarrhea. Like any other antimicrobial, it can also cause diarrhea. But what is special is we can utilize this side effect also. Like when we actually want to increase the GI motility, we can use this kind of drug. And the macrolides can be used for condition like diabetic gastroparalysis. Like if due to diabetes, if gastric motility decreases, we can stimulate motilin receptor to stimulate the motility. Similarly, it can also be used in paralytic ileus. Now, second adverse effect is allergy. It can be used by it can be caused by any antimicrobial drug. So even macrolides can cause allergy. C is for cholestasis. Cholestasis jaundice can occur. But the special point to remember over here is this is not the side effect of all the macrolides. This cholestatic jaundice, it is mainly caused by a group of macrolide, that is erythromycin. And even not all sort of erythromycin causes cholestasis. It is basically seen with erythromycin acetylate. Okay, so only erythromycin acetylate can cause cholestatic jaundice, and the risk is very much high during pregnancy. Therefore, only erythromycin acetylate is contraindicated in pregnancy. Now, what is special to remember over here is, as such, macrolides are safe in pregnancy. So, as such, only erythromycin salt and macrolides, they are safer in pregnancy. Only the drug which is contraindicated is erythromycin acetylate. And even erythromycin acetylate, they are not causing any kind of teratogenicity. They are just producing cholestatic jaundice in mothers. That's it. Now, next adverse effect, RO, means reversible ototoxicity. Now, most of the drug that causes ototoxicity, they can cause irreversible ototoxicity, like drugs such as cisplatin, vancomycin, and aminoglycoside. These all drugs can produce irreversible ototoxicity, but macrolides, they can produce reversible toxicity. So this is a special side effect. They causes reversible ototoxicity. Now, other important thing to remember regarding macrolides are tacrolimus. Now, you must have heard about the drug tacrolimus. Yes, it is an immunosuppressant drug, but you will be surprised to know that tacrolimus, these are also macrolides. See, macrolide, it is a chemical structure. Same chemical structure which is present in other macrolides like erythromycin, clarithromycin, and azithromycin. And same kind of chemical structure can be seen in tacrolimus too. So basically, macrolide group of drug, they possess both antimicrobial property as well as immunosuppressant property. You can say like tacrolimus, they produces immunosuppression. Okay, so these are basically used for immunosuppression, autoimmune diseases, and transplant. Whereas other drugs such as erythromycin, clarithromycin, and azithromycin, they can be used for their antimicrobial activity. Now, among these macrolides, there are also some kind of differences in azithromycin basically and other macrolides. And the important differences are these azithromycin, they are very long acting, whereas other macrolides, they are relatively short acting. Other macrolides, they have got microsomal enzyme inhibition, whereas azithromycin, they do not inhibit microsomal enzyme. And because they do not inhibit, they do not produce any kind of alteration in microsomal enzyme, they have got fewer chances of drug interaction, whereas other macrolides, they have got higher chances of drug interaction. Okay, so this was all about the macrolides. Now going to our next topic, that is clindamycin. Now regarding clindamycin, mechanism of action is same as that of macrolides, like after formation of peptide bond, translocation occurs, and basically clindamycin are the MCQ drug which will basically inhibit the translocation. Next, we have got three important points to remember regarding clindamycin. First thing is, clindamycin is secreted in bile, therefore they are saved in renal disease. Secondly, it was the common cause of pseudomembranous colitis, that is PMC. In fact, due to this reason, we avoid to use clindamycin. And thirdly, major indication of clindamycin is for anaerobic bacteria. Okay, so these are the three important points regarding clindamycin. Now, the major uses of clindamycin can be remembered from the mnemonic CAP. C is for cocci. Clindamycin is useful in cocci like cephalococcus, streptococcus kind of organism. A is for anaerobic bacteria. Clindamycin is effective against anaerobic bacteria. In particular, it is used in case of acne vulgaris. Okay, so whenever there is acne, we have got a ointment, a cream of clindamycin. So it can be used for treating acne vulgaris, the pimple kind of thing. Even it is used in lung abscess caused by anaerobic bacteria. Lastly, P means parasites, and the important parasites against which we can use clindamycin, they can be remembered as PMT. P is for pneumocystis, M is for malaria, and T is for toxoplasma. Okay, so these are the important uses of clindamycin. Our next class of drug is quinpristine, and it has also caused the same mechanism of action, that is, it will, these are the MCU kind of drug that will inhibit the translocation. Okay, next, this drug, that is quinpristine, it have got its best friend, means quinprestin is always given in combination with a drug known as dalfopristin, and both the drug combination chemically they are known as streptogramins. Okay, and these streptogramins they are basically indicated in VRSA, that is vancomycin resistant staph aureus. Now these streptogramins they are given in VRSA, but the thing to remember is they are not the drug of choice in VRSA. They are one of the drug used in VRSA. As such, drug of choice for VRSA is daptomycin, but when daptomycin is not used or any other problem occurs, means we can use streptogamin in that place. Okay, so this is all about quinprestin. Our last class of drug is aminoglycosides. Drugs coming under the category of aminoglycosides are streptomycin, gentamicin, tobramycin, natilomycin, neomycin, capreomycin, kanamycin, and amikacin. Now, as a group, special point to remember about aminoglycosides are not effective orally. The reason is that they are not well absorbed. Now, because they are not effective orally, we need to give aminoglycoside by injections only. Next, their aminoglycosides are mainly effective against gram-negative bacteria. Like if you compare penicillins, they are effective against gram-positive bacteria, right? And these aminoglycosides, they are basically effective against gram-negative bacteria. Even they are also effective against pseudomonas also. Next, aminoglycosides are not effective against anaerobic bacteria. Now, what might be the reason is basically the aminoglycoside they require oxygen. So, any bacteria that uses oxygen, means whatever aerobic bacteria are there, via oxygen the aminoglycosides can enter into the bacteria and they can inhibit the bacteria. But in case of anaerobic bacteria, since they do not take oxygen, so aminoglycoside they cannot enter inside the bacteria. So, therefore, because of this reason, aminoglycosides are not effective against anaerobic bacteria. Next, aminoglycosides are cidal drugs. These are the only protein synthesis inhibitors which are cidal, and all other drugs in this category, that is protein synthesis inhibitors, are bacteriostatic. And the reason we know, because aminoglycoside will produce cidal protein, there will be misreading because of which a wrong protein will be produced, and that protein is usually a cidal protein. So therefore aminoglycosides are the bactericidal drugs. Next, special side effects of aminoglycosides are they produces ototoxicity and nephrotoxicity. Even they are producing neuromuscular blockade. Now, among ototoxicity, they are of two types. Like we have got auditory ototoxicity as well as vestibular ototoxicity. Now, over here, amikacin can cause maximum auditory toxicity, that is auditory ototoxicity, whereas streptomycin can cause maximum vestibular ototoxicity. Now, vestibular system is to maintain the stability of a person. So, therefore, this streptomycin will affect the stability of the person. Lastly, nephrotoxicity is produced by neomycin. Okay, and neomycin not only it will produce nephrotoxicity, plus it will also produce neuromuscular blockade. Okay, so this is all about aminoglycosides, and this was all about my today's video regarding protein synthesis inhibitors, the antimicrobial that will produce action by inhibiting the protein synthesis of the bacteria. I hope you people found this video helpful. Thank you for watching.